this lecture, we are covering glycolysis, which is covered in Chapter 15, Sections 1, 2, and 4 of Voigt, and Pratt. Specifically, we are focusing on the steps in the glycolytic pathway and the enzymes involved and how this pathway is regulated by controlling these enzymes. Here are the learning objectives or outcomes for this content. We will spend 2 class meetings discussing this content in more depth. In our first meeting, we will look at how the regulation of enzymes controls the flow of metabolites through glycolysis using inhibitors and allosteric activators. This will allow us to interpret the impact of different Km values in real scenarios, describe how isozymes and allosteric effectors play important roles in the regulation of pathways, and allow us some practice in communicating information using graphs. In our second meeting, we will explore the role of the liver in glycolysis, gluconeogenesis, and in the body. This will allow us to explore our understanding of the roles of muscle and liver in glycolysis and gluconeogenesis, and should also further enforce understanding of isoenzymes and their role in regulations of glucose, synthesis, and catabolism. These activities will also give us some practice in thinking critically about the link between the macroscopic, or organ function, and the microscopic, or metabolism of glucose, giving us perspective in glycolysis and gluconeogenesis at a different level. So in order to come to class prepared, the ability to meet these learning objectives listed here on the slide should make the in-class activities more impactful and meaningful. Glucose is the major source of metabolic energy in most eukaryotic cells and is the starting material of glycolysis. It typically appears in the blood from the breakdown of dietary or stored polysaccharides and enters into the cell by carrier proteins, where it is then transported into the cell cytosol, the site of glycolysis. Once glucose is brought into the cell, glycolysis can be initiated. In this overall 10-step pathway, we see that glucose is broken down into 2 molecules of pyruvate and also nets 2 molecules of ATP, 2 molecules of water, 4 protons, and 2 NADHs as byproducts of the reaction. Glycolysis can be divided into 2 stages, as indicated in this figure. In the first stage, 2 molecules of ATP are consumed to phosphorylate and cleave glucose into 2 molecules of glyceraldehyde 3-phosphate. In the second stage, we convert the 2 molecules of glyceraldehyde 3-phosphate to 2 molecules of pyruvate, releasing enough energy to make 4 ATP molecules and 2 molecules of NADH. In the overall process, we have a net gain of 2 molecules of ATP and 2 molecules of NADH for every 1 molecule of glucose that enters glycolysis. At the end of glycolysis, pyruvate is then further oxidized through aerobic respiration to generate more ATP through the citric acid cycle and the electron transport chain. Under anaerobic respiration, when oxygen levels are low, pyruvate is converted to lactate through the enzyme lactate dehydrogenase to regenerate the NAD+ needed to continue glycolysis. If you're interested in studying the role of LDH in more depth, consider my Advanced Biochem course, where we extract LDH from heart tissue and study the kinetics, structure, and thermodynamics of this enzyme and analyze its roles in cancer cell proliferation. Another route for pyruvate in yeast cells is alcohol fermentation, where pyruvate is converted to ethanol and carbon dioxide. This is the process in which we make beer and wine, and why beers and wines with higher gravities or higher alcohol concentrations can taste less sweet, because more of the sugars from the malts or the grapes have been converted to ethanol. The side product of carbon dioxide also gives beers its natural fizziness. Now, let's look at glycolysis step by step. In the first step of glycolysis, glucose is phosphorylated by HEXOKINASE in the presence of magnesium and ATP to make glucose 6-phosphate. The phosphorylation of glucose prevents transportation of glucose out of the cell and is the first committed step of glycolysis, meaning it is irreversible. The phosphorylation of glucose to yield glucose 6-phosphate is an endergonic reaction, requiring 15 kilojoules per mole of energy. By coupling this reaction with the dephosphorylation of ATP, which releases approximately 30 kilojoules per mole of energy, we now have a net release of 15 kilojoules per mole of energy, making this reaction highly favorable and spontaneous. As I mentioned previously, hexokinase phosphorylates glucose using ATP only in the presence of the cofactor magnesium. Magnesium is required by kinases such as hexokinase because it is used to help shield and stabilize the negative charges of the phosphate groups on ATP, allowing the gamma phosphate to be more accessible for nucleophilic attack preceding the phosphate group release and transfer. In step 2, an isomerase, phosphoglucose isomerase, converts aldose glucose 6-phosphate to the ketose fructose 6-phosphate. This reaction is readily reversible, meaning the delta G is near equilibrium. However, conversion of G6P to F6P exposes the carbon 1 and allows for phosphorylation in the next step of glycolysis. The rapid phosphorylation of fructose 6-phosphate in the preceding steps shift the equilibrium in favor of fructose 6-phosphate production. While the exact mechanism of PGI is still not completely understood, it has been proposed that it acts as a general acid-base catalysis through an enediol intermediate. In step 3, fructose 6-phosphate is phosphorylated at C1 to make fructose 1,6-bisphosphate using phosphofructokinase in the presence of magnesium and ATP. Again, this phosphorylation is endergonic, but through coupling with the dephosphorylation of ATP, we yield enough energy overall to make the reaction favorable. Because this coupled reaction is so exergonic, it is also irreversible and is the next committed step of glycolysis, and it is also the site of glycolysis regulation. We have found that PFK1 can be inhibited allosterically by high levels of ATP and citrate, and is also allosterically activated by high levels of AMP. Therefore, if ATP is high and energy is not needed, glycolysis will be turned off through the inhibition of this step. And if energy levels are low and AMP concentrations are high, we can increase metabolic flux or the rate of glycolysis to help generate more ATP. Accumulation of citrate, being an intermediate in the citric acid cycle, is also an indicator that ATP needs are low and will also slow down or inhibit glycolysis by inhibiting PFK1. By inhibiting PFK1, we will see a buildup of fructose 6-phosphate, which will shift the equilibrium back to accumulate G6P, which can then inhibit hexokinase and prevent initial phosphorylation of glucose and completely stop glycolysis in the cell. In step 4, fructose 1,6-bisphosphate is cleaved into an aldehyde, glycerolaldehyde 3-phosphate, and a ketone, dihydroxyphosphate, with the enzyme aldolase. Unlike the isomerase reaction in step 2 that is near equilibrium with a DG of 0, this reaction is actually thermodynamically unfavorable, but it still occurs rapidly as the products of this reaction are quickly removed in the next step of glycolysis. In step 5, DHAP is isomerized into GAP by triose phosphate isomerase, and while this reaction is reversible, this reaction favors production of GAP as GAP is used up in the next step of glycolysis. At this point, we have finished the first stage of glycolysis, in which glucose is phosphorylated to G6P, then isomerized to F6P, phosphorylated again to make FBP, and then cleaved to make GAP and DHAP. In stage 2, as GAP is consumed in the next step of glycolysis, DHAP is isomerized to make more GAP. Remember, in this stage of glycolysis, 2 molecules of ATP are consumed using kinases, which are also the committed steps in glycolysis and therefore are points of control, where the enzymes that govern these reactions can be inhibited or allosterically activated to increase or decrease metabolic flux through glycolysis. Step 6 of glycolysis starts stage 2 of glycolysis by phosphorylating GAP to make 1,3-bisphosphate glycerate. using the dehydrogenase reaction governed by the enzyme glyceraldehyde 3-phosphate dehydrogenase. Because this is an oxidation reduction reaction coupled with phosphorylation, NAD+ and inorganic phosphate are used in the reaction, and NADH and a proton are byproducts of the reaction. This reaction is exergonic due to the oxidation of the aldehyde group by NAD+, which releases enough energy to drive phosphorylation of GAP. As an interesting side note, the enzyme in this reaction, GADPH, is a tetramer of 4 identical monomeric subunits, and each subunit can bind GAP, NADH, and phosphate, allowing this enzyme to reach high catalytic efficiency and turn over compared to enzymes that can only perform one reaction at a time. In step 7, we transfer a phosphoryl group from BPG to ADP to make 3-phosphoglycerate and ATP. This reaction is governed by the kinase phosphoglycerate kinase, and since it is a kinase, it requires magnesium. This reaction is an example of substrate-level phosphorylation in which ATP is produced through phosphate transfer utilizing a kinase instead of through oxidation reduction reactions that occur in the electron transport chain, which we will study later on in this term. By coupling the reaction of the carbon-1 phosphoryl bond, which releases about 50 kilojoules per mole of energy, to forming the phosphoanhydride bond between the beta and gamma phosphate groups of ATP, which requires 30 kilojoules per mole of energy, we have a net release of 20 kilojoules per mole of energy, making this reaction thermodynamically favorable and spontaneous. This release of energy also helps the previous slightly endergonic step in which GAP is phosphorylated to make BPG occur in the forward direction as well. In step 8, we have the freely reversible conversion of 3-phosphoglycerate to 2-phosphoglycerate using the enzyme phosphoglycerate mutase. This reaction occurs as a 2-step addition-elimination reaction and sets us up for the next step in glycolysis to make phosphophenylpyruvate, which has a much higher energy release upon dephosphorylation than 3-PG does. Dephosphorylation of 3-PG would only release about 12.6 kilojoules per mole of energy, where the dephosphorylation of PEP releases about 61.9 kilojoules per mole of energy. One of the intermediates in this reaction is 2,3-bisphosphate, the molecule we discussed in oxygen transport using hemoglobin and myoglobin. Recall that 2,3-BPG binds and stabilizes the deoxygenated state of hemoglobin, allowing for more efficient transfer of oxygen from hemoglobin to myoglobin. This is just one of the many examples in which we see that intermediates from one pathway can cross over into a different cellular pathway. If you take a moment to stop and consider this specific example of pathway crossover, it is actually quite a brilliant bit of evolutionary efficiency. If a cell is trying to actively generate ATP using glucose in aerobic respiration, the cell needs to ensure that sufficient oxygen is being released in order for the citric acid cycle and the electron transport chain to occur. If there is a buildup here in glycolysis, it may be an early indication of oxygen shortages in the cell. Therefore, a buildup of 2,3-BPG can then induce more oxygen released to allow the citric acid cycle and the electron transport chain to continue operating, and therefore allow glycolysis to continue as well. In step 9, 2-phosphoglycerate is dehydrated to phosphophenylpyruvate via enolase. PEP has a much higher phosphoryl group transfer potential than 2-PG due to its enol phosphate group instead of the phosphate ester. Hydrolysis of the enol phosphate group from PEP releases about 60 kilojoules per mole of energy. In step 10, the final step of glycolysis, PEP undergoes a substrate-level phosphate transfer to ADP via pyruvate kinase to make pyruvate and ATP. Remember, for every molecule of glucose, we receive 2 molecules of PEP, and therefore generate 2 molecules of ATP in this step for every molecule of glucose. As we have seen with the other kinases governed reactions in glycolysis, this step is also irreversible. Reviewing stage 2 of glycolysis, we see that we generate 2 molecules of pyruvate, 4 molecules of ATP, and 2 molecules of NADH. If you recall from stage 1 of glycolysis, we consume 2 molecules of ATP per molecule of glucose, meaning in the end we have a net gain of 2 molecules of ATP per molecule of glucose when we combine both stages. So now that we have generated 2 molecules of pyruvate per glucose, what happens to pyruvate? Well, we have 3 possibilities. Pyruvate can continue through aerobic respiration using the citric acid cycle in order to generate more ATP, NADH, and FADH2, which can then go through the electron transport chain to generate even more ATP. Or, if oxygen levels are low, pyruvate is reversibly converted to lactate to regenerate the NAD+ needed to continue more rounds of glycolysis. In yeast, we have another pathway where pyruvate is converted into ethanol and CO2 in alcoholic fermentation. Again, the process we see in beer and wine. If you recall from our previous in-class assignments on the Introduction to Metabolism, we were analyzing the steps within the metabolic pathway to predict which steps were used to generate metabolic flux, or the steps used to control the rate at which the pathway flowed, and which steps were the sites of enzymatic control. You may have noticed that these steps are actually usually one and the same, meaning the steps that slow down or speed up reactions are often the irreversible steps that are able to be controlled by enzymatic inhibition and/or activation. For instance, in glycolysis, we see these key steps are irreversible are the steps 1, 3, and 10. All the steps that are governed by kinases and facilitate phosphate group transfers with large negative releases of energy. If we look closer at these steps, we can see how glycolysis is regulated by controlling the catalytic rate of these specific enzymes. HEXOKINASE, the enzyme in step 1 of glycolysis that phosphorylates glucose to make G6P, is inhibited by high concentrations of ATP and G6P. PFK1, which catalyzes step 4 of glycolysis, where fructose 6-phosphate is again phosphorylated to make fructose 1,6-bisphosphate, is inhibited by high levels of citrate, a byproduct of the citric acid cycle, and ATP. It is activated by fructose 2,6-bisphosphate and AMP. Pyruvate kinase, the enzyme in step 10 of glycolysis that makes pyruvate, is inhibited by acetyl-CoA, which is the end product of the citric acid cycle, and ATP, while it is activated by fructose 1,6-bisphosphate and AMP. In addition to the direct control of enzymes within the glycolysis pathway, the cell can also control the metabolic flux of glycolysis through the release of hormones, glucagon and insulin. Glucagon is a hormone that is released by pancreatic cells when blood glucose is low. It activates the phosphatase activity of the enzyme PFK2, which reduces the level of fructose 2,6-bisphosphate. Since fructose 2,6-bisphosphate is an allosteric activator of PFK1, reducing its concentration in the cell reduces the metabolic flux of glycolysis. In contrast, the release of insulin by pancreatic cells when blood glucose is high activates the kinase activity of PFK2, which increases fructose 2,6-bisphosphate concentrations in the cell, thereby increasing the rate of PFK1 in glycolysis and increasing the overall metabolic flux of glycolysis. Throughout this video, we have summarized the 2 stages of glycolysis, examined this pathway step by step, and explored how glycolysis is regulated through inhibition, activation, and hormones. Over the next 2 class meetings, we will explore this pathway in more depth and look at how various methods of regulation affect intermediate buildup and how this buildup can direct intermediates to other metabolic pathways in the cell. The activities in class will allow us to explore our understanding of glycolysis and gluconeogenesis, and should also further enforce understanding of isozymes and their role in regulations of glucose synthesis and catabolism. These activities will also give us some practice in thinking critically about the link between the macroscopic and the microscopic perspectives in glycolysis and gluconeogenesis. So make sure you are ready to take what you have learned in this lecture to apply it to our activity in class to help deepen your understanding beyond simple memorization. I will see you in class.